Hello, everyone. Welcome to our episode 3 of our lecture series on single-cell high-throughput data science. Today, we are going to talk about data modeling for single-cell RNA-seq data and how I'm going to do normalization, and we'll mention imputation method. Through the last 2 lectures, I hope you have some understanding how the single-cell experiment will be conducted and what platform people will use normally, and how I will use Cell Ranger as a software solution provided by Genomics to produce this single-cell RNA-seq expression matrix. This matrix is also called UMI count matrix. UMI stands for unique molecular identifier. Each number here is integer greater than 0, greater or equal than 0, and indicates the gene expression level of the gene. In this particular example, this matrix contains over 6,000 genes and their expression in 444 cells. So this part of this matrix, you can see the name of the genes in the row. And then you see the columns indicate the cells, okay? So what to do if you have such a matrix? You can use Excel sheet to do some basic analysis, but normally you will use the computer language. So for the tutorial, in the next couple of lectures, I will use MATLAB, but any other language like R and Python will also applicable here. And if you look at the expression matrix, this from the real data, you see there are some characteristics about the single-cell RNA-seq matrix, expression matrix. First, you see the gene has a different expression level. So this gene, ISG15, have relatively higher number in each of the cells expression. So overall, this gene expression level is higher than the most of other genes you see from this view. And also, you can see a lot of zeros here. That's one of the most important characteristics of the single-cell RNA-seq expression matrix. Those zeros indicate there's no gene expression of this particular gene, for example, in cell number 2. Okay. So once you have a matrix like this, the first thing you want to do, you may want to have a sum of each rows. Okay. If you sum all the values in this row, you have get the total expression summary of this gene across all the cells you have measured here. So you want to do this for all the genes. If you have a gene, we don't have that example here, but if you have a gene with a sum completely 0, it means particular gene's expression has not been detected in all the cells you measured. You probably want to get rid of this particular gene, because this gene expression level is so low, it's undetected across all the cells you sampled here. So you want to get rid of some of these rows. Some gene maybe not absolute 0 in their sum of the expression, but once the expression level is too low, you may also want to get rid of them. So that's a very basic filtering. to get rid of those genes, no or extremely low expression level. You may want to do the same for the cells. If you have a cell and you sum its expression across all genes, if you have 0 or very close to 0 or extremely low compared to other cells, you may want to get rid of them. So I'm going to use a smaller example to show you how this will be done. Assume you have done this very basic filtering. You get rid of the genes, have very low expression. You get rid of cells, have very low expression, and you obtain a filtered UMI matrix. An example like this. Then you may want to do follow the routine. It's something called library size normalization. Okay, I would like to see this is a very naive approach. There are some problems with it, but it's common practice. Almost everybody is using this. This have a particular reason to do it, and their assumption I will mention. I will certainly will mention how can you do this more sophisticatedly. It's an advanced model of statistical models. But let's start from very simple idea. We assume if you don't do this, for example, if you don't do library size means the sum of the column of all the detected rate across all the genes. You sum it. That's the library size for each cell. But if assume we don't do it, and then you may want to summarize the rows, and this gene expression is at this level, and this have a same expression level here. This looks like it's a direct comparison of gene expression. However, if you sum the columns, you will see some of the cells have very low library size. Okay, because some of this is only 2, and the sum of these cells have very high library size. We want to normalize this because the assumption here is a typical assumption. Total gene expression, that's total gene expression, sum all the gene expression level across all the genes. The total gene expression per cell should not differ much between cells. Okay, that's a general assumption. The observed variation, the variation that's the difference between this library size across cells. This variation in the sequencing dips. So library size is a library size. It's also proportional to the sequence dips. We assume it's due to the technical bias. So if everything is the same, and we assume we should sequence same amount of rates across all the cells, you expect all these numbers should be the same. So that's an assumption in the naive library size normalization. Let's see how we can do it. It's pretty easy, and all you have to do is to divide these values by its total. So I will divide 1 by 7, 2 by 7, because this cell have a library size of 7. I will do the second cell. I will divide 1 by 5, another 1 by 5. For the third, do the same thing. For this large library sized cell, I will divide 2 by 10, 1 by 10. So if I do this, I will obtain a normalized expression matrix. So this matrix is the same as the previous, it is divided by the corresponding library size. So after this normalization, I would like to see the library size is used as a scaling factor. You rescaled each expression level of each gene. And then if you sum this normalized matrix, all the cells will have the same library size. It's normalized. After the normalization, you can start to measure the gene expression, overall gene expression for different genes. Okay, so the rank of this looks like different from your previous this, okay, before normalization. We will see, okay, this after scaling scaled by the library size, this normalized matrix will be ready to use for downstream analysis, such as compare mean expression of the gene between genes. Again, I'd like to emphasize this naive assumption, although we use this a lot. There are some problems associated with that. Okay, so library size normalization is not something new. It has been used for normalized bulk RNA-seq, the traditional RNA-seq data analysis. So I'm going to go through this paper mentioned. So this is an unpublished manuscript called Beyond Library Size, a field guide to next generation sequence normalization. So library size normalization belongs to a category of so-called global scaling normalization method. So they rooted from the traditional bulk RNA-seq analysis. So we have a raw data matrix. And in this example, the two samples is from one from patient, one from control. So this is traditional RNA-seq comparison. There are two samples. You can think about this as a single cell 1 and cell 2. Single cell RNA-seq, you just have many different samples. But the principle is the same. So we have different ways to do normalization. One is library size normalization. So that's, and then there's one called RPKM, which is specified for bulk RNA-seq. I will mention this. And then there DE-seq. DE-seq is differential expression analysis package. They use a specific normalization method. So I will go over 3 of these independently, so that you have some idea about what we are talking about. Okay, so library size is a column sum of this. So for this sample, you have a library size 120. For this sample, you have library size 60. So library size normalization, you just need to multiply a factor, scaling factor for this factor, for this sample. So you end up with the same size of a library after you rescaled every numbers in this column. Okay, so that's the library size normalization. RPKM stands for rate per kilo million per million rates. So the difference between this and the normalization by library sizes is consider the length of the gene body. Okay, you have a longer gene, then you expect to have a more rates to be mapped in there. So this normalization, RPKM normalization, is not applicable to single-cell RNA-seq because single-cell RNA-seq, the detection method will not be sensitive to the length of the gene. They only use hybridization of the 3' poly A tail and to the 10X genomic GLB barcode. There's no mapping process, so there's no RNA from other part of the body of the gene, so we don't have to use this normalization. The only difference between this RPKM with library size is consider the gene length. So we don't have to worry about this for single-cell RNA-seq analysis now. Okay, the last normalization method introduced in this paper is DE-seq. So this is slightly special. It's found the majority of gene and assume majority of the gene do not appear to change expression. So this assumption may be biologically more feasible, and because you don't expect the major change for every gene change between these two samples. So DE-seq will assume gene 2 and 3 between two samples, there are no expression change. So they will be using to compute scaling factor. Okay, instead of using all genes expression difference to compute the scaling factor, only use those assume there's no changes. There are some advantages about using this kind of normalization. Okay, let's go back to single-cell RNA-seq analysis. What's the problem with global scaling normalization? The problem has been illustrated in this paper, and assume we have a different type of cells. And for example, macrophages is much larger than lymphocytes and neutrophils. And then the assumption of the library size equality uniformly distributed across all the cell types will not hold at this situation. So before normalization, you see the library size for data from macrophages is much larger than the other cell types. So if we still naively assume they should have the same level of library size, we will normalize it by the same factor, scaling factor, and force them to be equal. In this case, all the other genes expression in this library size matrix will be changed accordingly. And then this will skewed or biased all the gene expression level and produce spurious differential expression for particular genes across different cell types. The solution for this is more complicated than what I can explain clearly here. Basically, you need to model the data. Okay, you want to do the imputation, and then you want to cluster in different cell types at the same time. What that means is you have to learn from this matrix, and for each cell, you want to find the statistic description of the gene expression across genes. So if you assume there are 3 clusters, then you will have to use 3 sets of these modeling parameters to define them. Then you group cells into different clusters. At the same time, you want to filter in the parameters to estimate the each gene's expression level and correct it by individual scaling factors. So this process depends on the statistic modeling of data. It's much computationally intensive. That's why although the naive library size normalization has a lot of problems, but still we just cannot use this more sophisticated method because that will take very, very long to compute. But for us, we need to understand what the modeling of data, what's the principle behind that. So I'm going to go back to very basic probability distribution to data. So that's all the statisticians are working on. So they collect data and try to use statistics to understand the data and generate the model. So model is a distribution of your data and the frequency of the what the value of data you will observe. Once you have this model, then you will be able to use a probability model to regenerate data. So that's what we call generative models. So I'm going to give you an idea about what generative model can be used. It can be used to simulate data. Okay, so if I see, I'm going to use a generative model to simulate a set of data. In this particular example, I'm going to simulate the hat of the man from Italy and the man from Sweden. So we have some statistics among the average body height of the men in these 2 countries. So because I have this model on my mind, so I can start to generate random samples. For example, that will be something like this. I will write values around this mean because I know the parameters to define this model. Okay, sometimes I have higher values. Sometimes. Okay, so to generate 3D the data, I'm going to use my parameter in my model. Okay, you can see the difference. So I'm writing this random numbers, but not completely random. It's around the parameter space that's defined by the generative model. It's on my mind. So on average, I will assume the average of this is 1.81 meter, and here is 1.76 meter. Okay. So if I want to simulate single-cell RNA-seq data, I have no idea now which model I can use. There are many, many different statistical models, distributions. If I don't understand my data, I will not be able to generate a very reasonable simulated data set. So that's the purpose of the modeling. So we need to understand which statistical model, which distribution will be best to be used to randomly generate RNA-seq data, which close or very similar to observation. Now let's think about how we can generate this model. So how can we start from data and then from data to get the probability distribution that best for described the data we observed. Once we have that, we can simulate the data. So we collect data points and generate histogram, which is the frequency of particular range, and then the data point will be fall into this range. That's a histogram. So once we have a histogram, we can rebuild the generated model for distribution of the probability of this. So that's the purpose, the modeling process. And the distribution function, we can have a different distribution functions. Okay, so normal distribution is most commonly used for statistical purpose. Okay, so as I said, the human head, weight, salary, etc., can be modeled very accurately by using two parameters. One is the mean, and another is the variance. So we all know this very well. However, for single-cell RNA-seq data, as you can see, they don't follow the normal distribution, so we cannot use normal distribution to model it. So after a few years study, now the statisticians have realized the negative binomial distribution will be the proper model to model the single-cell RNA-seq data. Okay, that's the definition of the mass function of the probability, mass function of this distribution. That's expected mean and like variance distribution. So let's see what this distribution is about. We require two parameters to define a distribution, that's probability of the success and number of trials before you can get the so X will be numbers of trials you have to be able to get R times success. So that's the meaning of this distribution. I will give examples to see how and why we're using this to model the single-cell RNA-seq expression number of reads. So I borrowed the documentation of MATLAB function for negative binomial random number generator. So this function will generate a random number follow the negative binomial distribution. See, it requires two parameters, R and P, to give the random number. Okay, the random number will be an integer greater than 0. So the R variable indicates corresponding numbers of success, and the P is the probability of success in a single trial. And in this documentation, they also provide an example. See, how are you going to use this function to simulate, to generate the random numbers of the output. Okay, in this example, they say, suppose you want to simulate a process that has a defect probability of 0.01. This is 1% of defect probability. So this is the P parameter here. And then the question will be, how many units, that's how many R, the value of R is. So how many units met quality assurance inspect before finding 3 defective items. So that's the 3, that's the parameter. Okay. And then for this function, you see, I want to ask to generate 6 times, to simulate this process 6 times. So let's create a vector by 1 and 6. So the number here is 1 of the simulation. So you need to try this 496 times. You will get 3 defects before you get 3 defects. Okay, plus 3 because you tried 493 times without success, and then you have 3 times success. So add 3 in. And another times you may have to try 142 times, and then so that's the random number which generated based on this assumption. So let's replace this example with the scenario which we use single-cell RNA-seq experiment. So I change the defect probability as a detecting probability. So assume we have a capacity using 10x genomics technology, and we can detect expression level of gene if you have a 1% of detecting power. So that means if you have a 100 mRNA, then there's 1. You probably will generate 1 UMI. Then the question we will ask is, how many RNA molecule of the gene might be expressed? That's the original expression level of the gene. So how a gene, the real gene expression level has to be in order to given UMI count of the gene is 3 has been detected. So this question is, if we observed in the expression matrix a gene expression level is 3, and then we know the detecting probability is 1%, then what's most likely the original expression of this gene will be? So you can simulate, see, if I have a 3, UMI equals 3, and detecting power 0.01, and then the real gene expression number could be either this or that. There's an average around these vectors. Okay, so that's the example that how are you going to simulate the original gene expression average by given UMI using non-negative binomial distribution. Okay, so in the real global scaling normalization, and people, statisticians also want to consider a lot of other factors which can give you the variation and introduce the difference in the gene expression level observed in UMI matrix. You want to consider amplifying factors. This will be defined for each individual cells to estimate. You have a capture or reverse transcription fraction, another parameters, dilution for the parameters, sequencing dips. So all these factors can be modeled using generating models. And then the whole purpose, you want to specify, estimate a scaling factor for each individual cell one by one, okay? To correct the uncertainty among this. So I think I'm running out of time, and the whole purpose of this talk is not to give you any detailed analysis of exactly how I'm going to do, but to give you the idea about naive library size estimation, normalization, and also you know the problem is and how are you going to use more sophisticated normalization method and what's in the information in there. Finally, I want to mention the imputation method is also, a lot of them is based on the model, okay? So a lot of imputation method. Imputation means you want to fill in the unobserved information, and also oftentimes you're doing the normalization and imputation at the same time. And we have a expression data from a different cell types and maybe different correction expression single cell RNA-seq technology. You can do basic normalization and transformation QC, and then there are many imputation methods can be used, and there are smoothing based and deep learning based, and this is a matrix decomposition based, but a lot of this also are based on the modeling of data. Try to understand how the data should be generated correctly. Once you have those parameters, you can start to fill in those gaps in the observed matrix. So once you finish the imputation, you may have a better power in the downstream applications to do the clustering, trajectory analysis, the differential expression, network. That's an assumption. Okay, the rest of the time I'm going to use MATLAB to show you how the basic command line of MATLAB to be used to the library size normalization, and some of these imputation tools have been incorporated into the MATLAB. I'm going to switch to the MATLAB environment I'm using. Okay, so what you see right now on the screen is the interface of MATLAB environment. So MATLAB is a scientific computational tool and the environment for computational engineering computing. And what you can see on your top is the menus and the buttons. Create transcripts, write your code, and here you have the current folder and the files in the current folder, And here you have the preview of your files, and your example data, for example, is here. So it has this matrix X and has gene list. In the middle, this comment window, and then on the right, this workspace to tell you what's right now in the memory of the computer and in the workspace environment. So this is interactive language, so you can use it as a calculator to compute the basic mathematics operation. And the MATLAB is designed for manipulate matrix. So there are a lot of functions. For example, random generator, it can generate a 3 by 4 random number. Okay, so the matrix. And all the operation is based on the matrix operation applied to matrix itself. So if you plus 2 for all element in this matrix, you can just plus 2, and every single values in the matrix will have a 2 plus. I'm going to load example data from text genomics, and to show you how easy you can use MATLAB to do this library size normalization. So in this file, I have example text genomics data file. I will double click to load the data. So you see, once I load this data in, this example data in my workspace, I have a variable called X. So X is where your UMI expression data is contained. It includes 8,842 genes in 1,567 cells. So what is this matrix look like? I can double click, and you can see the matrix. There's the expression value, UMI count. Okay, just like what you see in the Excel spreadsheet. You see some of the gene may have a higher expression than others. So gene 561 seems all values is higher than other genes. So this is a highly expressed gene across all the cells. And if you want to know what's the name of the gene, you can double click the gene list and try to find the gene in the row 561. So let's double click this. Then you can see the name of gene and the 561. The name of that gene is TMA7. So this gene is highly expressed. Okay. So how are you going to normalize it? So before normalize it, maybe we want to take a look of what the matrix look like. I'm going to use a function called img C, and X is just too big, so I'm going to select the top 100 genes in 200 cells. See this sub-matrix, what it looks like. Okay, that's the matrix. It's a subset of the whole expression matrix. And you can see a lot of zeros, and the blue zeros. So this is a very sparse matrix, and some of the values have really high, and up to 120 in this particular gene separation in this cell. Okay. So now I'm going to compute the library size from X. I'm going to clean my command line, and then the library size, I give a name, variable name, lab size. All you have to do is to compute the sum. So this gives you the library size of all the cells. So you see, once I run this function, to create a new vector called lab size with 1,567 values in there. Those are the library size for each cell in this X matrix. Okay. And how I'm going to normalize to get the normalized UMI matrix. I'm going to use X divided by the lab size. That will give me a normalized matrix. Okay, so in MATLAB, this kind of high-level language, that's the command you want to use to compute the library size. And then you just divide all the columns by their corresponding library size, and then you obtain the normalized matrix. So you see, the normalized matrix has the same dimension as the original matrix, the same size. And then double click, you will see the value are not integer anymore because it's been normalized by divided by library size accordingly as a scaling factor. And then those are the normalized library size. Oftentimes, this value is too small. So you may want to make the value larger by multiplying it by a constant. So in this case, I use 10,000, and then you change the value, so give you a reasonably large expression values. Okay, in the 1.0. Okay, now I can validate, see after normalization whether the library size will be the same, because the whole purpose of the library size normalization is to rescale the library size for each cell accordingly, and then at the end, across all the cells, we have the same library size. So if I do sum of X norm, and then the library size 2, so I recompute library size across normalized matrix. And now you can see they all the same values. The value is the 10,000 across all the cells. Okay, and then that means we have already normalized all the cells in this way. All right, so if you want to know how to use MATLAB, then you can, let me clean the environment, and you can tap doc. That will bring you to the documentation of MATLAB, and there are many tutorial examples and example scripts you can run and get started. You will learn the how you quickly get started language function to tell you the syntax, how are you going to define the matrix, how do the indexing. Yeah, we can, for example, click this start. You have a desktop basics. I just showed you where the command line window, there's a workspace variable window, and then you can look at the definition, how are you going to create array, create a matrix in the language. So that's the basic. And also, if you installed a package called single cell gene expression analysis toolbox, there this is the library, the function library I developed for the single cell RNA-seq analysis. There are demonstration script. You can, for example, click and run these examples. In this example, I will start by reading into example data, which has already been prepared in the example data folder. And then see what we can do with some basic manipulation of the data. Select genes, so we have some quality control to get rid of lowly expressed genes. Basic filtering. And then, because I'm using two examples, I want to get rid the intersection of these genes. And then I can take a look what the matrix look like. On your X and Y, the two matrices, left and the right of this yellow line. And then this function doing the basic normalization using tSNE method, as I mentioned in the lecture. So this is the matrix after normalization. You can use the simple naive library size normalization. You get slightly different results. Okay, because after the library size normalization. And you may also want to do this log transformation. So after log transformation, you start to see the difference because they rescaled and give you the more closely, the values close to each other, so you start to see the variability. Otherwise, you'll only be able to see using the color bar. And the magic is one of the smoothing-based imputation tool. And in this toolbox, we can run this to get imputated matrix. So you see, after that, a lot of noise has been cleaned up, and assumably the downstream analysis will be more meaningful after the imputation. There are other normalization method, imputation method. There are methods for correct for the batch effects. Okay, that's about the basic manipulation normalization for the initial original UMI matrix. And there are other scripts which will demonstrate how I'm going to do other downstream analysis. For example, next lecture, we are going to talk about visualization, dimension reduction. Okay, for this, for now, let's go back to the slide. So I summarized what we have just showed using this very simple MATLAB command line to do library size normalization. Once you load that in, basically 1 to 2 lines of code will give you a normalized matrix. If you want to scale it up a little bit, and then you multiply the whole matrix by a factor. Normally people use 10,000. Okay, that's all for today, and looking forward to see you again next lecture.